Hello everyone. Today I am talking about the cancer therapy. In cancer therapy, there are four different type of cancer therapies there: chemotherapy, radiotherapy, surgery, and biotherapy. These therapies are used on the basis of the type of cancer, cancer's location, and its severity. Here, these methods are being used either radiotherapy along with the chemotherapy, or chemotherapy after the surgery, or biotherapy along with the surgery. Like that, different combination of methods can be applied for the cancer therapy. Right? Usually, in case of the surgeries, whenever some residual cells remains in that particular cancer site, they go for the chemotherapy as well. So here I am discussing first the biotherapy. Biotherapy, the word itself indicates it is something related to the our body itself. Means the therapy by using our body's method or our body's product. Right? That's what called biotherapy. Usually we are preferring it for the very bulky cancers and cancers which are clinically apparent. In this, what we are using, they are the therapeutic use of any biological substances. Means biological substances are going to be used. These biological substances can be anything. The substances derived from your body. They can be some kind of cells. They can be certain kind of extracts from your body or the human body. So these are given a special term called as biological response modifiers, BRM, because ultimately they are something which belongs to your body. When they go into your body, they modify the body's response, right? So that's what ultimately the biological responses have been modified. So they are modifying your own biological responses. Means this is a kind of very good technique by which your your body's response will be modified, right? It will be either it can be enhanced or it can be suppressed as per the need. And because in our body all kind of responses are there, you have seen it in previous slides that oncogene is there, proto-oncogene is there, right? So like tumorogenic factors and anti-tumorogenic factors, all kind of factors are there in our body. As per the need, they activate it. So here also we use these BRMs to modify those factors and their responses. BRM work through the diverse mechanism. Very important to understand these points. First point is BRM augment the host defense through the administration of cells. One thing I told you, this biological substance can be the cells, can be the natural biological products, or the synthetic derivatives of that, right? And what they do, they are of as effectors or mediators of an anti-tumor responses. So ultimately what you want in case of cancer, you want your anti-tumor responses or anti-cancer response from the body should be stronger. So these cells or the natural products will enhance these anti-tumor responses. Point number 2, increases the individual's anti-tumor responses through augmentation or restoration of effector mechanism, or decrease a component of the host reaction that is deleterious. It means what? They augment the or they restore the effector mechanism, right? By which these things can be controlled. So and that is in case of cancer, that methods are not really working good, right? They are distorted. So body will these agents will restore that thing, okay? As well as they will subside and they will suppress those reactions of the body which are deleterious, right? Which are enhancing the cancer condition. So these components are very much effective in these two methods, right? They will restore the effector mechanism, as well as they will decrease or suppress the deleterious mechanisms. So this way these biological response modifiers have a great role. Okay, so ultimately what they are doing, they are controlling your own body's response. They are not bringing something from outside. They just go inside and control your own body's response towards a particular situation. What is the situation here? That situation is a tumor. That situation is a overgrowth of the cells. That situation is one mechanism in your body is overriding the normal mechanism. So ultimately, you have to control those overriding mechanisms. You have to control those mechanisms which are not following your body's system. Okay. Point number 3 here: augment the individual's responses using modified tumor cells or vaccines to stimulate a greater response or increase tumor cell sensitivity to an existing biological responses. What does it mean? Here, the agents BRMs are either they are the modified tumor cells, means tumor cells itself genetically modified, injected back to the body where they started responding in a different method, right? Which in other term we can call it as a vaccines as well, right? So these are the agents which go into the body, injected into the body, and what they do, they stimulate a great response. Means earlier they were not strongly responding, but now they are when they are modified, now they are responding good, right? Or they are increasing the tumor cell sensitivity. Okay. Next point is decreases transformation and increases differentiation or maturation of tumor cells. What does it mean? They decreases the transformation. Ultimately, they, you know that when one cell divides and forms another cell, they go for various processes, right? They go for the proliferation, then they go for transformation, right? And the differentiation. So these differentiation means, first thing, a normal cell converts into a cancer cell, that is a transformation. So either these agents will control that transformation process, or they will control the differentiation process, or they will control the maturation process. Means the tumor cell is formed but should not get matured, so they'll control that maturation process as well. Next point is interfere with growth promoting factors and angiogenesis inducing factors. Very important point. Last point I told you in the last slides, that angiogenesis, where the new blood vessels are formed. So this is a once the one tumor start forming their own blood vessels, their growth will be very rapid. So there are we need certain agents, and in our body itself we have those agents. We need just to modify them so they can act strongly against that angiogenesis process what the tumor is doing. As well as growth promoting factors, there are, you know that there are various factors which are actually uh, acting as a growth promoting factors. They are acting as a supportives for your cells, right? So they are controlling those factors as well. Next point is that decreases or arrests the tendency of tumor cells to metastasize. You know very well how dangerous it is. When a tumor starts spreading, right? When a tumor cell start moving from one place to other place, from one organ to other organ by using the blood or other channels, and it reaches there and establishes or implants itself and start growing as a new tumor. So metastasis is one of the very dangerous situation, right? And in that way controlling will be very difficult. So these agents can control that metastasis process as well. Next point is to increase the ability of the patient to tolerate damage. This is very important. This is a very different kind of innovative idea what they do, because we are giving chemotherapeutic agents. Most of the time patients are not dying because of the cancer effects, but they are dying because of the drugs effect, right? Because their body is not able to tolerate those drugs effect. So what these response modifiers do, they will make the patient more endured. Their endurance will be high for these deleterious effects, these adverse effects of these drugs, and body will be able to cope up with those responses and will be able to deal with the consequences of those drugs and behave in a better way. Next point is use biological molecules to target and bind to cancer cells and induce more effective cytostatic or cytocidal antitumor activity. Now, for very well that when a body decides that cell to give a death to a particular cell, that responses, if that responses get modified, then only the cancer growth is happening. So we can modify that response and we can enhance the cytocidal activity of that. Here there are some examples given: biologicals and biological response modifiers. First category of these are called as immunomodulating agent. Means your immune system will try to fight back with these transformation where the normal cell converts into the cancer cell. So once your immune system get enhanced or get more strengthened, then they fight back and try to get back those cells into the normal position, normal condition. Here we have some examples. There are some endotoxins, some immune RNAs are there, which are maybe genetically modified cells. T cell growth factors are there. You can find various examples. Now the here number 2, interferons and interferon inducers. Number 3, thymosins. Number 4, antigens. And number 5, effector cells. You can see the whole effect cells, the complete cells can be used as an agent. Either they are macrophages or the natural killer cells or cytotoxic T cells, LAC cells, T helper cells. So the entire cell will be act as an agent and will be injected into the body to enhance those body's responses, right? Same way antigens are also there, which can be the molecular vaccines or can be anything. So like that, these are the biological response modifiers. Fifth one is the lymphokines, cytokines, growth maturation factors. So huge list of these agents are there here, as well as there are some miscellaneous approaches which can also be employed. What these miscellaneous approaches are there: viruses, infection of cells, right? So here viruses are being used to do that particular job. Nowadays the viral viruses are being modified as a treatment agent, and these viruses can do a lot many good things if they are genetically modified, as well as bone marrow transplantation and reconstitution. So these are also things are there which act as an BRM to strengthen the therapies. Now I'll talk about the chemotherapy, which is the main part for us here. Usually, when we talk about the chemotherapeutic drugs, you remember when we were talking about the antibiotics, there we always discuss about the adverse reactions of antibiotics, and we found one adverse reaction which is very common in some antibiotics, that calls cytotoxicity, right? So now that time we say these antibiotics are not good, those who are having high cytotoxicity, but those agents, those antibiotics are very much useful here in case of the chemotherapy, because what we need to do, we want the cytotoxic activity of the cell, of the agent. So we have to understand the cell cycle kinetics for understanding the cell growth, because unless until you don't know how a normal cell converts into a cancer cell, so what is going wrong into their cell cycle kinetics, what is going wrong in the cell cycle physiology and process. So if you understand properly that what is actually the cell cycle kinetics, how the cell cycle growth, cell cycle goes on, how the cell cycle being controlled, how the cell cycle being divided in various phases, which are the enzymes, which are the hormones, which are the factors which are taking part into it. Once you understand this whole particular process, then you are able to control this process. Once you are able to control this process, it means you are in the process of making a drugs which can control. And once you can control the cell cycle, when you control the metastasis, when you control the cell cycle, when you control the mitosis, you are able to control the overgrowth of the cells. When you are able to control the overgrowth of the cells, that's here you are, right? That's you are on the way, or you are almost near to find a medicine or for a treatment for this cancer. Okay, so cell cycle kinetics, it is one of the very very important understanding. In cell cycle, I think you have gone through cell cycles before, where you can find the cell cycle have the various phases, right? Where you have the different phases, and all these different phases do the different jobs. In the next coming slides, I will discuss about that. Now, the cell cycle. Cell cycle, the cell cycle is that process by which a one cell divides, multiplies, and differentiates, go for differentiation for the assigned work for that. So this cell cycle is not that simple which is look like. There are various checkpoints, controls, regulators by which only the cell cycle been allowed. Okay, the cell cycle consists of 4 phases. One is called the pre-synthetic growth phase 1 or G1. In this growth phase 1, synthesis of DNA is not happening. That's why it's called pre-synthetic growth phase 1. That's why it's called G1. Second is DNA synthetic phase. That's why it is denoted as S. S means synthesis. So DNA synthetic phase means in this particular phase DNA is going to be synthesized. Third is pre-mitotic growth phase 2, means in this, before the mitosis begins, this phase comes. That's called the growth 2, G2 phase, right? In this, the remaining growth will be taken care of, right? Then it goes into the mitotic phase, or that is called M phase. You know very well that what is happening in the mitosis, you already have studied before. So mitosis again will be divided in next further phases, where you can divide it as the prophase, metaphase, telophase, anaphase, and by that cell goes into the division. One more phase which comes in between here, that's called the resting phase, right? Or which is actually the sub phase of G1, that is called as a phase 0 or G0 phase, right? In this, what happens? Cells that are not preparing for cell division, there are certain cells which cannot because of certain reasons, right, they are not allowed to go into the cell division process, because once they go into cell division process, they will multiply and they will make their own copies. So sometimes they are not properly formed, they have certain kind of damages, so body won't allow those cells to go for the further division, right? And those kind of cells will be kept as in the subphase or G0 phase, right? G0 which is before the G1, because once the G0 phase completed, G0 phase, if the G0 phase is not there, after mitosis again they can go into the G1 phase. That's why after mitosis there will be a proper check that all those cells which are divided are correctly divided or not. If they are not correctly divided, they are not allowed to go into the G0 phase. Like that, this whole cell cycle will be divided into the pre-synthetic growth phase, DNA synthetic phase, pre-mitotic growth phase, mitotic phase, or resting cells, or G0. So we can call it as a G0, G1 phase, S phase, G2 phase, M phase, and G0 phase. In the coming slides, I will show you the diagram of the cell cycle, by which you can understand better that which part of the cell cycle is being controlled by what, right? So in this process, why we need to understand the cell cycle? Because various drugs are acting on the various phases of the cell cycle. Here we can see like one diagram which is showing G1 phase, which is a blue color. G1 phase is given. I'll begin with this. And G1 phase needs the highest time period to complete, that is 6 to 12 hours, right? What is happening into this G1 phase? G1 phase, RNA and protein synthesis happens, but no DNA synthesis happens here. Once these synthesis, RNA and protein synthesis going on, after that there is a restriction point. Restriction point is that particular checkpoint where it has been checked that the cells which is being passed, which has been done the RNA and protein synthesis correctly done or not. If any problem, that restriction point won't allow the cell to go into the next phase, that is the S phase, because there is a little dangerous. Once it go into the S phase, the DNA will start synthesizing, right? So that's why it controls it here itself. But with the cells which are correctly formed, they are allowed to go into the next phase, that is from G1 to S phase. Now S phase, which is the second highest, second biggest one, 6 to 8 hours it takes to complete it. In this, actually the DNA synthesis happens. That's why it is one of the very, very important phase. It doubles the amount of DNA in the cell. RNA and protein also synthesized. So most of the synthesis work is happening here. You can later find out most of the drugs are there which act on this particular phase to stop this particular process. Now we go to the next phase, that is the G2 phase, which comes after the S phase. G2 phase, which is the third largest, which takes 3 to 4 hours to complete the process called G2 phase, and G2 phase has no DNA synthesis. It only synthesizes RNA and protein synthesis again. After the G2 phase, once all things have been synthesized properly, the cells has been transferred to M phase, that is the mitosis process. Now it go into the mitosis process that takes 1 hour, and this mitosis process or M phase, where what is happening there? Nuclear divisions and nuclear division, cytokinases, cell division, and then by this 2 daughter cells will be formed. Like that, M phase will be completed. And you remember M phase has the prophase, metaphase, telophase, anaphase. Like that, it is divided into it. After the M phase completed, when the mitosis got over, the cells will go into the again into this cell cycle process, into the G1 phase, and they'll start dividing. They'll go again, repeat the same process again for the next set of cells. But what is happening here? After the M phase, certain cells which are, as I told you, they may not be very good. They have some issues. Our body will take a decision which cell to go into G1 and which cell not to go into that. So what will happen here? It will go into the G0. Those cells will go into the G0 phase, right? G0 phase, terminally differentiated cells withdraw from cell cycle. So these cells will be withdrawn from the cell cycle, right? And they will go into the G0. It is also called as re-entry point. A cell returning from G0 entry. A cell returning from G0 enters at early G1 phase. It means now if later body decides that the cells which are in the G0 phase and need to come back into the cell cycle again, in that case, this is the point they will enter into the G1 phase. So a cell returning from G0 enters at early G1 phase. Then these cells will enter into the G1 phase in the early point, and then they will continue the cell cycle by itself. So here you can see very well that cell cycle and its whole process is very very important, and that's why we have certain drugs which are acting on the various phases of the cell cycle. They'll not allow those cell cycle to complete, and when the cell cycle will not complete, then what will happen? The cell cycle that will stop at one particular phase, and the new cell or the new cells will not be divided. So by completing the G1 phase, S phase, G2 phase, and M phase, the whole cell cycle is get completed, right? And in this way, once the cell cycle completed, then it means one cycle is completed and the new cells have been formed. Here, in the next coming slides, I will discuss about the various classifications, and in that you will understand these drugs which are acting on these particular phases. This is one more diagram which talks about the similar thing like G1 phase, S phase, G2 phase, M phase, where mitosis is happening, DNA synthesis is happening, and growth is happening in that particular phase. So like that, G0 is also been shown. Now it is also another diagram which talk about the G0 phase, G1 phase, S and G2M phase. Here some drugs are also been written, those drugs which are actually inhibiting these particular phases. If you see the S phase specific, there are some drug name as cytosine, arabinocyte, hydroxyurea, and S phase specific are sex mercaptopurine, methotrexate, and same way you can find in the M mitosis phase there are vincristine, vinblastine, paclitaxel, like that. So there are various drugs which are acting on these particular phases to inhibit that, to suppress that, to stop it, anyway to control the cell cycle process, right? And in the classification again you will find these drugs under that category. Means category of cell cycle specific drugs, means the drugs which their mechanism is based on the cell cycle inhibition. So they are not like inhibiting the all this phases of the cell cycle, but they act on particular phase of the cell cycle and will try to inhibit that particular phase. This next diagram will tell you little more things which is very very important here. As I told you, when a drug, when a cell moves from the one phase to other phase, that cell needs a lot of control, right? And this process, as I told you, that from G1 to it goes to GS phase, then G2 phase, it's not that simple. Here lot many things are happening, lot many controls have been there. Now I'll tell you what is that. These are called as the cyclin proteins, right? Cyclin protein name given because they are involved in cell cycle. So cyclin protein controls the entry and progression of cells through the cell cycle. Cyclins accomplishes their regulatory function by activating the constitutively synthesized proteins called cyclin-dependent kinases and CDKs inhibited by proteins such as P16. A very important point here. What happens, so whenever cell cycle progresses, right? Suppose that now the cells which are in the G1 phase want to go into the S phase, then there will be a G1 cyclin protein. The G1 cyclin protein get activated, and this will activate G1 cyclin dependent kinases that call G1 CDK. Now once these CDK get activated or synthesized, now this CDK will take this particular cycle forward into the S phase. Now once it will move from G1 to S phase, the G1 cyclin will degrade. So when G1 cyclin will degrade, now you need another cyclin protein there, then another cyclin protein will come, that's called the S cyclin protein. S cyclin protein will synthesize another kind of CDK. What is that CDK? Called as SCDK. That process will keep on moving, and after the S phase it will go to G2 phase, right? It will go to M phase. So what will happen then? M cyclin will come. Now the M cyclin, before that S cyclin degraded, then M cyclin will come. Now the M cyclin will activate MCDK, right? Then MCDK will come. Then after, once it will go from M to G1 again, then the M cyclin degraded, then the M again G1 cycle will activate, right? Like that it is happening every from when the cycle moves from one phase to other phase, right? Whenever cycle moves from one phase to other phase, before that it is compulsory that a one cyclin protein has to be get activated, and that cyclin protein will activate or will synthesize that particular cyclin dependent kinase, right? You know the kinases, role of the kinases you have seen in all the various pathways, and there you can understand how these kinases act, right? So that kinase will form. These kinases are actually sequential process, right? They activate one by each other. They activate to some other kinases, then other kinases like that move and give the signals further. So here these CDK will get activated, and once the process is moving to other one, it is compulsory that the previous cyclin has to has to degrade, because once that cyclin will degrade, that particular CDK will stop functioning. Okay, and because that cyclin needed for that CDK, and that CDK is needed to regulate that particular process in that cycle, that phase itself, right? So now once this cyclin degraded, that CDK also inactivated. Now for the next step you need another cyclin. So there it will come that new cyclin, and that new cyclin will activate the new CDK, right? And then that process happens. Then after that again it will destroy, it will degrade, and again it needed for the new cyclin and the new CDK. So like that these new CDKs are coming in. What happens, like if suppose that if you inhibit these CDKs, right? So there is a one protein called as P16. So suppose that this particular gene, which is the P16, if it is activated, and once it is activated, it will inhibit CDK. Once it will inhibit the CDK, actually it is stopping the cell cycle process. Okay. So it will stop the cell cycle process, and that's what actually you need. It means there are already a preset system in your body where P16 is available, and once the cell cycle get completed, the P16 will activate and inhibit the CDKs, and the cell cycle will stop. But in cancer case, what happens, these proteins or these genes are not properly acting, and in that case you need certain agents to strengthen them. Okay, Here is one more specific example involves CDK1, which controls the critical transition from G2 to M. This is just one example given that how G2 to M something is moving. Here if you see, again same thing, CDK1, then it will CDK1 moves and comes here and activates cyclin B, right? Synthesized beginning at G2. Now cyclin B, it is where it's synthesized, just beginning of the G2 phase. So this cyclin B will bind with the CDK and it will inactive, it's kind of inactive kinase. Now then CDK and cyclin B, once they comes together, they get phosphorylate. Once they get phosphorylate, that CDK get active, and this CDK get active, and the process of transition from G2 to M phase will happen, right? Here you can see, and the phosphorylation happens by various ways: protein phosphorylation, mitotic spindle formation. So what will happen here in this particular phase, because of the phosphorylation happen, and CDK got active, so the process of G2 to M phase transition starts. So what are the things are happening there? Mitotic spindle formation is happening, nuclear lamina depolymerization is happening, chromosome condensation is happening. So these are so many processes are happening in this particular phase. Okay, so go through with this particular cycle and you can understand that how what is the role of the cyclins and the CDKs. In the examination, there will be a one single question on cell cycle or the cell cycle regulator, like that. Now after cell division, cyclin B is degraded, as I told you, whole this process, and here various checkpoints also I told you, G1 to S and G2 to M, like that various checkpoints will come. Points at which the cell checks whether its DNA is sufficiently replicated and all mistakes repaired before progressing, right? G2 to M phase. So, what's happening there? And because before that, in the S phase, DNA is already synthesized. So before the cell is going into the mitosis, it is very important to check all the things and find out that things are correct. So that's what the cell will verify. Cell will put that means cell will be verified by those checkpoints, and they will ensure that all the DNAs are sufficiently replicated, right? And any mistakes, any damage into that DNA is repaired properly. Once they're ensured, then they allow that cell to go for mitosis, because mitosis, once they go wrongly into the mitosis, what will happen? Then this cancer kind of situation can happen, right? And wrong cells or the cells which are not abiding the body's rule can be formed. Failure to adequately monitor the accuracy of DNA. You can ask me one question here, like when this much controlled mechanisms are there in your body, then why cancers happens? Okay, these many controls, these many checkpoints body is taking care of, even though cancer is happening. Why? That's why it is one of the rare diseases, right? It's not happened to everyone. Every one of us are involved or undergo into the mitosis process every day, right? All our cells are dividing, all our cells are proliferating by using the same cell cycle, by using the same what called the factors, by using the same checkpoints, by using the same cyclins, right? But we all are not the cancerous. Reason is that only rarely it happens, right? Rarely it happens. What rarely happens? Failure to adequately monitor the accuracy of DNA replication leads to the accumulation of mutations and possibly malignant transformations. Means DNA has not been verified well because of certain reason, because of some inability of the body's mechanism. And then what will happen? A wrong cell will be formed, or maybe the mutated cell has been formed. And these mutations will bring out the various diseases, including tumors. As an example, when DNA is damaged, suppose that by ultraviolet radiation, the tumor suppressor protein, which is TP53, is stabilized and induces the transcription of CDKN1A, a CDK inhibitor. What is that? Means suppose that a body has found that one DNA is damaged. Now immediately we cannot allow this cell to go for the mitosis. So what body does? Body immediately activates the tumor suppressor protein. Tumor suppressor protein, which is name is TP53. What this will do? This will activate CDKN1A. What is this CDKN1A? This is a CDK inhibitor. As I told you in previous slide, for completing the cell cycle, what you need? You need a CDK, right? To inhibit that process, what you need? You need a CDK inhibitor. So like that, body behaves. Whenever body needs to stop that process, body will activate its own system. By that, tumor suppressor proteins will activate, and they will activate CDK inhibitors, right? There are certain drugs which we have, they are also acting as a CDK inhibitors. This arrests the cells in G1 or G2 until the DNA can be repaired. At that point, the TP53 level falls, CDKN1 diminishes, and the cells can proceed through the checkpoints. What does it mean? It means when the process of repair is going on, the cells have been stopped, the whole cell cycle process has been kept on the hold or standby. Once this repaired process completed, then these cells will be given back, the process has been given back to go, and they start the process again, right? And that is also happening in case when there is no repair possible. Then what will happen? Then it is allowed that the cells must go for the apoptosis. If that is not happening, then that is a case the wrong DNA will go into the mitosis and make the wrong cells, and these wrong cells may generate various diseases like cancers. If the DNA damage is too extensive, as I told you, TP53 will initiate a cascade of events to convince the cells to commit suicide, right? You might have heard the word suicide for the human beings. When human beings decided not to survive, they go for suicide. Same thing here, cells. Body is taking the decision to kill one of its cell. So how it is happening? Because every individual cell having its own system of certain things. So body will convince by sending some messages, by sending some chemical informations, right, to that particular cell, that to commit suicide, and then that kind of cell will die and go out of the process. Many of the most effective cytotoxic agents act by damaging DNA. As I told you, because we have a lot of drugs which are acting for the damaging the damaging the cell and by damaging the DNA. Their toxicity is greater during the S or DNA synthetic phase. Why they are highly active during this phase? Because these are the two phases where the S phase where the actually DNA synthesis is going on. These drugs name for very common drugs name as vinca alkaloids and taxanes. These blocks the formation of functional mitotic spindle in M phase. What these drugs are doing? They are actually blocking the formation of a functional mitotic spindle in M phase. You remember your mitosis class where you have understood that one mitotic spindle formation happens during the M phase, right? During the metaphase, and that will be blocked by these vinca alkaloids and taxanes. Conversely, slowly growing tumors with a small growth fraction, for example carcinomas of the colon or lung cancer, often are less responsive to cycle specific drugs. What does it mean? It means because as I told you, these drugs are acting highly on the cell division process or the synthetic phase process, but there are certain group of cells which are dividing very slow. it means the process of division is very slow, the process of synthesis is very slow, and in that case these drugs are also not very much effective. They are acting slowly on them. Okay. Anti-cancer agents have variable pharmacokinetic and toxicity in the individual patients. The causes of this variability are not always clear, and they often may be related to inter-individual differences in drug metabolism, drug interaction, or bone marrow reserve. So where different drugs gives the different effects to the different patients because of the inter-individual variability, the patient's immune system, the patient's endurance, this all depends. And because this all depends, the drug's pharmacokinetic is also depending on the different patients, right? Some patients can tolerate better, some cannot tolerate better. Some patients, the pharmacokinetic of these drugs are common, normal, some are not normal, like that.